Laura Forero es profesora asistente en The OU State University, donde investiga cómo los virus manipulan las respuestas de interferón y cómo estas dinámicas impactan la inflamación y la progresión de la enfermedad. Su trabajo integra virología, inmunología y biología de sistemas para identificar nuevas estrategias terapéuticas que restablezcan el equilibrio inmune frente a infecciones virales agudas y crónicas. Démosle un aplauso a la doctora y adelante.
Perfecto, muchísimas gracias. Para mí es no solamente un placer, sino yo creo que en estos momentos la cumbre de mi carrera profesional, haber nacido y crecido en Barranquilla y volver después de 25 años a estar aquí en la Universidad del Norte presentando enfrente de ustedes. Así que gracias al comité organizador, gracias a ustedes por estar aquí hoy sábado. Sé que hay cosas chéveres que hacer aquí en Barranquilla, pero ajá. Me encantaría decir como Carolina que les voy a dar toda la presentación en español, pero el trabajo no lo hice yo, lo hizo mi estudiante y no sería capaz de comunicar efectivamente, ¿sí?, lo que se hizo, pero también terminar a tiempo. Y como hoy es sábado, sí, hay cosas que hacer, me demoraría como 3 horas tratando de pensar en cómo decir cosas, sobre todo que estamos abriendo un campo nuevo en el laboratorio y aún estoy aprendiendo hasta en inglés. Entonces, aquí vamos a hacer un cambio y aprecio que estén aquí conmigo y, ajá. Bueno, en todo caso, so, let's go. My lab is interested in really understanding the dynamics that set the host ability to detect viral infections, synthesize interferons, and respond to interferon stimulation. This response starts with detection of pathogens that leads to the synthesis of interferons. And this interferon response then induces the expression of interferon-stimulated genes. So we've heard from Carolina, we've heard from Sibel, being talked about, that are the antiviral effectors. A subset of these interferon-stimulated genes is actually cytokine and chemokine genes that are necessary for the recruitment of cell mediators, immune cells, that are going to go on to amplify this antiviral and this pro-inflammatory response. The reason we need to understand the molecular mechanisms that are dictating the dynamics, the magnitude, the time in which this response happens, and the magnitude with which this response happens, is that there's a very fine window of protective inflammatory responses that is elicited by any given cytokine, but particularly by interferons. And when we look here at harnessing the protective efficacy of interferons, we're not only talking about protection against viruses, but also other pathogens, as well as maintaining tissue integrity, for which interferons actually play a baseline sort of role in homeostasis. When we lose this fine balance, not only are we more susceptible to infection, which is why we're here talking about virus responses, but also we surpass this protective inflammatory threshold and we get into detrimental, deleterious inflammatory responses that actually increase the immunopathology of viral infections, but can also lead to tissue destruction. This is probably best exemplified by the immunopathological roles of interferons outside of the context of infection, which is what we see in autoinflammation and autoimmune diseases that actually have a very strong interferon signature. So we've talked about interferons here a lot over the past 3 days, but I want to make a case for the fact that interferons are not the same cytokines. Interferons are actually a complex of 20 different cytokines that we group within 3 different families, the type 1, the type 2, and the type 3 interferon receptor. And this grouping really has to do with the ability of these cells to interact with cognate receptors at the cell surface, which are quite distinct for each interferon family. It's important to understand that viral infection across all cell types will induce types 1 and 3 interferons. This is our cell-intrinsic interferon response. But the ability of cells to respond to type 1 and type 3 interferon is actually restricted by the expression of the high-affinity subunit for the type 3 interferons or interferon lambdas to epithelial cells. And this is why the primary antiviral defense in mucosal barriers are type 3 interferons or interferon lambda. We see the same spatial compartmentalization of interferon responses in that the secretion of type 2 interferons is actually mediated by our specialized immune cells. These are the only cells capable of producing type 2 interferons much later in infection. But not only is the biology of interferons dictated by their spatial compartmentalization, there's also biological activities that are unique to each one of these interferons. Interferon, by definition, means it interferes with viral replication. That's what the molecule was named in 1957, right? When it was first discovered. But what we can see is that the induction of pro-inflammatory responses is really something that is a biological property shared by the type 1 and type 2 interferons, and not quite by type 3 interferons. So this was really intriguing to me. If the infected cell can make type 1 and type 3 interferons, and in mucosal barriers, you have a co-expression in an epithelial cell of both type 1 and type 3 interferon receptors, then what guides that biological difference? Why do we need two cytokine families that basically have the same property? And so we started this out by trying to understand then how the antiviral response to is guided by type 1 and type 3 interferons through the activation of latent cytoplasmic transcription factors such as the STAT and the IRFs. These are canonical interferon regulators. They translocate into the nucleus upon activation and drive the expression of interferon-stimulated genes. And so looking at using genome-wide transcriptomics in interferon-stimulated immortalized hepatocytes, which are cells that express both the type 1 and type 3 interferon receptors in humans predominantly, what we see is that both cytokines are capable of inducing a core transcriptional program that is associated with antiviral efficacy. But only type 1 interferons can induce the expression of genes that are associated with both the recruitment of lymphocytes and leukocytes to sites of infection and their activation. And we found that at the molecular level, this is actually guided by the differential ability of type 1 interferons, in this case interferon alpha and interferon beta, to promote the upregulation of the transcription factor IRF1, but this was actually much more muted in response to type 3 interferons, and it did not matter which type 3 interferon species we used. To prove that IRF1 was actually the mediator of these pro-inflammatory cytokine-chemokine expression patterns, we actually generated CRISPR-Cas9 edited cells where we knocked out IRF1, and we were able to show that we still, again, conserve the antiviral properties of type 1 interferons, but we lose these pro-inflammatory responses. We're kind of interested then, knowing that we now have an ability to sort of learn from comparing and contrasting the functions of type 1 and type 3 interferons, to learn more about how to modulate, how to harness, how to change the dynamic of that type 3 interferon response to make sure that our sites, right, that are in constant contact with pathogens, our skin, our GI tract, our respiratory tract, chock full of epithelial cells, how are type 3 interferons conferring protection, and how do we harness that protective efficacy? So the next thing that we started thinking about then was the fact that in a dish, when we study these responses, we can say, all right, at time 0, I'm going to stimulate with interferon, and I'm going to track my response over time. But that response is not quite mimicking what happens in an organism. Right, in an organism, viral infection is not just inducing interferon responses, it's inducing, we've seen organelle changes, we've seen, there's interactions with the environment, so we have to consider, right, that these responses can be modulated by co-stimulatory signals, which can be the presence of growth factors, stress responses, or cytokines that are co-expressed during viral infection. But we could still take our sort of reductionist approach where we can just treat it with interferon and start modulating what we thought would be co-stimulatory signals to try to identify how this would affect the induction of antiviral responses. And we focused on a signal here, the conventional MAP kinase family of immune regulation, because they're downstream of a lot of these pathways, right? When we think of MAP kinases, we're talking about mitogen-activated protein kinases, and they're activated actually by growth factors, stress, and other cytokines. So we figured having a well-characterized immune regulator, why not start looking at the crosstalk between antiviral responses and other immune regulatory responses? And at the time, the laboratories of Steve Boulant and Megan Stanifer had shown that amongst the conventional MAP kinases that are characterized by having a kinase domain with this TXY motif, that they could actually selectively modulate antiviral responses to stimulation with type 3 interferons, but it seemed like type 1 interferons were a little less tunable. We wanted to extend this study to understand how a fourth member of this family, ERK5, could actually be playing a role in contributing to changes in antiviral responses. The reason we were so interested in ERK5 is that it not only has kinase activity, here you see it has that conserved TEY domain, but I really care about transcription, and it turns out that ERK5 has a long cytoplasmic tail that's actually capable to drive nuclear translocation, interactions with other transcription factors, and further guide transcriptional responses. Now, ERK5 is also known to mediate a lot of inflammatory diseases and cancers, so it's an active target for therapeutic development. And so we're like, well, why not try to see what the consequences of modulating ERK5 would be to antiviral control? And what we saw here was that if we could take chemical inhibitors that have been developed in the field, this is in this case XMD885, we can block the kinase activity of ERK5 without affecting the upstream MAP kinase signaling cascade, and try to ask what would happen if we pre-treated cells and then chased with interferon treatment to the induction of interferon-stimulated genes. And we use here ISG15 as a putative interferon to demonstrate changes in interferon responsiveness, and we saw that there was a dramatic inhibition of ISG induction after stimulation with type 3 interferons in the presence of the ERK5 chemical inhibitor, but there did not seem to be any effect in the induction of ISG15 in response to type 1 interferon. Again, showing us that dynamic change that we had seen before, when treating cells, but also recapitulating what the Boulant and Stanifer labs had observed before. Now, the thing is that we didn't know what the mechanistic basis for this was. And so what this experiment and the study that is preprinted here and currently under revision has actually taught us is that we went from kinase inhibitors to making genetic knockouts of ERK5 too. So we make genetic knockouts in our murine epithelial cells, and we can see that what happens is that upon the loss of ERK5 kinase expression now, not just the loss of its activity, we get a decrease in the expression and the activation of pSTAT1, which is the primary effector of ISG induction downstream of type 3 interferons. But if you were keen and you picked up on the fact that in my graph at the very beginning, I also had STAT1 downstream of interferon gamma and type 1 interferon, then there's no real reason why this should have been a selective inhibitor of type 3 interferon responses. It should have affected all signals. And so this brings us back to thinking about work done by Chris Garcia's lab and Juan Mendoza when he was there, where they were actually, what they were attempting to do was to create a type 3 interferon superligand, right, where they could harness the efficacy of interferons by increasing the affinity of the type 3 interferons or interferon lambda to the interferon lambda receptor 1, and they did that here by creating a mutant they called H1. And here in this graph, in this kinetic graph, what you're seeing here by relative arbitrary units is actually the activation of STAT1. When you stimulate cells with type 1 interferon, here interferon omega, right, you see that it takes a lot lower doses of interferon to induce STAT1 activation, but that response actually is quite high, and it will eventually plateau. When you treat cells with interferon lambda 3, this is the same interferon we're using in our experiments, you see that it takes far more, a greater mass of interferon to induce STAT activation, but that also peaks itself, right? And even if you increase the affinity of the ligand to the receptor, you only change the kinetics, but you can never surpass a p-stat maxima. And we think then that this p-stat maxima is actually what makes interferon quite tunable. And to further highlight this, going back to our initial study where we were looking at how STATs and IRFs are differentially regulated by interferon, one of the things that we had seen was that the induction of STAT1 is quite dramatically distinct. So if we had normalized our doses to 30 minutes post-stimulation, we would get the same amount of STAT1 phosphorylation. The phosphorylation of STAT1 continues to increase over time. We never achieve that further increase in our type 3 interferon cells. The only way that we can modulate this is by actually overexpressing the interferon receptor, where we now try to equalize the interferon responses. You can see here that kinetically it's a kind of plateaued response, right? It's sustained over a much longer period of time, type 1 interferon response, but now we would get induction of IRF1 and we would go into an inflammatory state. What that experiment also showed us was that unlike the type 1 interferon receptor, which is an ISG, type 1 interferon receptors get induced by interferon treatments. The type 3 interferon receptor does not change during infection. And so it seems to be an evolutionary mechanism to be able to mount a localized immune response, but a response that will not supersede that inflammatory threshold that leads to detrimental inflammatory responses and potential immunopathology. So then I was thinking, right, I started my lab in the middle of the pandemic. I was alone. I could not bring people into my lab, so I spent a lot of time with my thoughts. And I was thinking, MAP kinase signaling, it's growth factor signaling, mitogen signaling, stress signals. Their role is really to modulate transcription factors that are found constantly in the nucleus or reside in the nucleus constitutively, right? This is a response that actually guides development of cells and cell basal function, even prior to infection and to cytokine stimulation. And so if these factors are actually guiding cellular development and cellular identity, how does cellular identity and development itself contribute to the ability of cells to respond to type 3 interferons? And kind of to prove if this was really the case, if there was something that we were missing, so our approach has always been to look at transcriptomes, look at gene expression, look at ISGs. This ISG was induced, this wasn't. That is very informative in being able to dissect pathways that might be activated or not, but what we miss is the ability to infer changes in the functions of some of these transcription factors, because besides these transcription factors that bind DNA in a sequence-specific manner, what we find is that resident nuclear factors are also enzymes that can modify histones in a non-sequence-specific manner. They're also epigenetic readers that can detect changes in modifications to DNA. And there are also transcription factors that might or might not be contributing to transcriptional regulation in a sequence-independent manner. So to try to see if modulating co-stimulatory signals, in this case we use XMD-treated cells, we actually took a mass spectrometry approach to not only look at things that were changing at the transcript level, but also proteins that were changing in their abundance when we actually inhibit kinases that are meant to actually keep cells running in their day-to-day life. And what we found here actually surprised us. So here in light blue, it's actually all the factors that were changed at a significant manner past a statistical cutoff. But when we started looking at the genes and what the proteins and what these proteins' functions were, we actually started noticing that there was a lot of proteins that actually regulate not only transcription of genes, but proteins that when you lose their function, actually are associated with developmental disorders, right? And in reading to the literature, right, because I don't have a big lab, I cannot validate every single one of those proteins. I do have ChatGPT, but it's not very good. So I could not actually take approaches to go in one after another. One of the things that we started noticing was that these factors, when affected, right, because they regulate chromatin, cause a series of diseases called chromatinopathies. And these chromatinopathies manifest predominantly as neurodevelopmental disorders, but there's also been a link to propensity to infection that has been missed because individuals with chromatinopathies have another plethora of clinical presentations that have masked and do not necessarily go into the hospital for management of their infectious diseases. They're usually being managed for episodes related to their neurological complications, right? But there was abundant evidence in the literature of this potential link. And so to do this, we actually started to focus on taking a clinically guided approach. We took a focus on one molecule here, and this is MECP2. What we found in our cells was that treatment with XMD, or inhibition of ERK5, and co-stimulation of interferon relative to interferon-treated cells, over here you would expect cells, proteins that expressed with the same level should have all aligned over here, but what we see is that we had a dramatic reduction in the expression of MECP2 protein, or a dramatic increase, actually, in the expression of MECP2 protein compared to interferon-treated cells. The way that we actually interpreted this in our experimental design is that this is a solubilization of the protein. This resides predominantly in the nucleus, and it's so tightly bound to chromatin that when you try to isolate this protein, you have to degrade all nucleic acid to be able to have a proper recovery. But we had not conducted our mass spectrometry that way. We actually had taken the entire chromatin fraction that was non-soluble, and we had discarded that before we did our responses. So we thought that this might have been potentially a loss of function of MECP2 in the presence of our inhibitor. And why MECP2 is actually quite interesting is that its name is actually methyl CpG binding protein 2, and it's a gene that's encoded in the X chromosome, important for brain development and neuronal function. Diseases associated with changes in the dosage of MECP2, both the high expression of MECP2 that's caused by duplication, leads to MECP2 duplication syndrome, and by the loss of copies or the loss of function of MECP2 results in Rett syndrome. You can see here that there's a sex bias in these syndromes, and this has to do with the fact that it's predominantly males that will have duplication of MECP2 because they don't undergo X inactivation, right? Females will, and so any aberrant amount of MECP2 can actually be taken care of by our epigenetic system. But females that lose MECP2 actually develop Rett syndrome. Males were thought to not survive the complete loss and the complete absence of MECP2. And so that's why we're now, with better detection, being able to diagnose males now with Rett and Rett-like syndromes. But what was really interesting is that both the duplication of MECP2 but the absence of MECP2 actually leads to a higher propensity of respiratory infections and a much more adverse outcome to infection. In the case of Rett syndrome patients, the pathogen that causes this has not been well described. There's only been one report early on from an international database of morbidities associated with Rett syndrome, but there has not been any ID follow-up to identify what these pathogens are. On the other hand, boys with MECP2, the pathogen has been identified, and it so happens to be a viral pathogen. In this case, it's actually flu B. And so we now have an opportunity, right, using MECP2 as our sort of test case, right, to try to identify whether inborn errors of development are actually also inborn errors of immunity that we have failed to actually characterize as such. And so why we, again, were so excited to look at MECP2 in this context is because MECP2 is a transcriptional regulator. It binds, like I said, to methylated cytosine, right? And cytosine gets methylated by DNMTs, and when MECP2 recognizes methylation, what it will do is it'll recruit repressive complexes and inhibit transcriptional expression. Now, cytosine gets oxidized by TET enzymes into 5-hydroxymethylcytosine as a primary step of demethylation. And TET enzymes then, this process will actually cause a switch from recruitment of the repressive complexes to activating complexes, and it acts as a positive regulator of gene expression. And everything that we know about MECP2 in depth, right, predominantly comes from literature based on neurons. But you can see here from this Western blot that while expression of MECP2 is indeed highest in the brain, it is actually a ubiquitously expressed protein. Where we look for it, we find it, right? We can look at it in cell lines, we can look at it in tissues, and when we look for it, we find it. Also, to make a case for DNA methylation as a regulator of immune responses, it is now an active area of research to try to understand how the writers and the erasers of DNA methylation regulate immune responses. But what we don't know is what all of the readers, including MECP2, are doing to contribute to interpreting, to one, help seed that response, but also to interpret that response. And so we now have an opportunity not only to understand whether there's really these vulnerabilities, but really add to the body of literature of understanding epigenetic regulation of immunology. The other thing that was really interesting is not knowing, right, what was the pathogen that these patients were presenting with, taking into account that duplication syndrome predisposes to influenza B, right? We went with thinking about what were the common causative agents of lower respiratory tract infections that were available to us, right? And so we had collaborators that were working with all of these pathogens, but we had expertise in influenza A, and now we could compare our responses and what we saw in the absence of MECP2, what has been observed and reported for patients with duplication syndrome. The other great thing that we could leverage here is that there's a mouse model of Rett syndrome that has been used to characterize the neurological complications of Rett that we could actually use to probe the consequences then, experimental consequences and controlled consequences of influenza infection. So we're looking here at murine lung lysates, and again, we can see very high expression of MECP2 lost in our MECP2 null mice. And the first thing that we noticed it when we infected these mice with influenza was that they were incredibly susceptible to influenza mortality with doses that were non-lethal in their wild-type counterparts, littermate controls. We did, again, genome-wide transcriptomics. This is what we do. We love it. We can learn a lot when we don't know anything. It's kind of like the best fishing expedition that you could have. And what we noticed was that following mice after days 2 and 6 post-infection, both our wild-type and our knockout mice, is that there's the expression of the host response to infection really doesn't kick in our knockout mice until day 6 post-infection. And we were curious about these 300 genes that are expressed early on upon infection in wild-type mice, not in knockouts, and we noticed that these 312 genes are actually genes involved with RLR signaling or viral recognition, here TLR RLRs, and JAK-STAT signaling pathways associated with cytokine responses tend to be antiviral. We looked at the expression of interferon-stimulated genes, and again, we can see that interferon-stimulated gene expression is not only delayed, but it's very muted in the knockout mice, and this was quite surprising to us. Intriguingly, when we're actually comparing, we're looking here, we did not see any differences at baseline in expression of tissue interferons. But when we're looking here at infected animals, in gray are wild types, in orange are knockouts, what we can see is that there's no differences in the production of type 1 interferons, but there's a dramatic decrease in the expression of type 3 interferons in infected lungs. And this is associated with a decrease in chemokine expression such as CXCL10, which is important for the recruitment of monocytes and lymphocytes that are necessary to clear viral infected cells. When further characterizing the responses that led then to a higher morbidity and mortality in our MECP2 null mice, we actually started focusing on genes, in two gene subsets that we noticed were quite significantly repressed in knockout mice and to a lesser extent in wild-type mice. And these actually corresponded with the expression of type 2 pneumocytes. These are the cells in the alveolus that actually can self-renew and can regenerate the alveolus. These are also the primary targets of influenza in the lower lung. And the first responders capable of making interferons upon infection. We also noted then an upregulation of genes that was most predominant in knockout mice of transitional epithelial cells and goblet cell hyperplasia, increasing mucus, that would signify an increase in mucus production, but also in signatures of cell types that are actually suggesting a potential metaplastic repair response in the knockout mice that is not occurring in the wild-type mice. And we can further see here, if we look at the whole lung response, that indeed, again, the expression of genes that are associated with inhibiting viral infection is delayed in knockout mice relative to wild-type mice, and what this results in is, increased damage into the lung here. You see more yellow responses in these heat maps, and an increased lung injury. And even though these responses are kind of evident in wild-type mice, what this injury response is capable of kicking in is a very robust fibrotic response that is not necessarily detrimental, but it's also the primary response that's required for the repair of the alveolus. We are actually missing this in MECP2 null mice, and so we think that what happens is that not only you have an extremely quiescent lung, you have a lung that cannot repair. Physiologically, the consequence of this, when we look at whole body plethysmography, is changes in lung function. So we're measuring here tidal volume. Tidal volume measures the amount of air that's taken in into the lung. We see that in wild-type mice, they're getting sick from infection, and the increase, right, or deeper breaths usually associate with establishment of hypoxic states that you would expect from tissue damage driven by the virus. But what you can see in the knockout mice is actually a completely different trend. We're not seeing the same pattern of respiration, right, where they're now taking not only shorter breaths, but shallower breaths. And this is actually a sign of both constriction of the airways, likely associated with this increased mucus production, which we're currently looking at for now histopathologically, but also of potential neurological complications and loss of autonomous nervous responses that regulate lung function. But what was most striking and most surprising, right, was the lack of expression of these 1,741 genes that are expressed by day 6 post-infection in wild-type mice that are not expressed in knockout mice. And when we looked at this, we saw that this actually correlated again with cytokine responses, but also with TCR signaling pathways, right? So T lymphocyte cells. When we look specifically here in this volcano plot at what these genes are, nicely here we have MECP2 as a nice gut check, but we're now seeing interferon gamma, granzyme, and perforin, suggestive of a loss of probably cytotoxic responses in the lung. And these are the responses that are necessary now, that are mediated by lymphocytes that infiltrate the cell. Remember, I told you type 2 interferons are not made by the primary targets of the virus. They're now made by the lymphocytes that infiltrate the infected tissues. And this is interesting, but my lab never studied type 2 interferons, and this is the reason why I cannot do my presentation in Spanish, because I would not know how to say any of these things, because I'm just learning them, right? But what was really, really interesting was going through the literature and going back and reading the body of work that has described this patient population and finding a study in 1999 that has never been followed up that suggested that patients with Rett syndrome have a lower number of circulating CD8 T cells, right? There was no mechanistic basis for this, there was no consequence associated to this, and we were like, maybe we actually have a great model to try to understand why this is happening. And so we hadn't really designed our first experiment very well, so our mice were dying by day 6 post-infection, and we know, this is the beauty of using a very well-characterized viral model, such as influenza, we know that during influenza infection, the peak of CD8 T-cell responses doesn't occur until about days 8 to 10 post-infection. So when we had tried originally to measure these responses, we were not really getting a nice dynamic range to be able to see differences. So we decided to take a new approach. We were going to take our wild-type and knockout mice and actually lower the dose of influenza to see if we could now extend the lifespan of these mice following infection, but also to learn a little bit about dose-dependent responses to infection, considering that, again, PR8 is a mouse-adapted virus. It's highly pathogenic in mice. So again, what we saw now is that the survival is greatly diminished in MECP2 null mice. Mice are making it up to day 10 post-infection. We've actually tracked them out days 15 because we want to start looking at lung repair responses, and we know that the mice that survive the infection will go on to live much longer. Again, you see by tidal volume that we're able to recapitulate disease. These lower doses don't cause quite as much damage to lungs, so we're losing that hyperinflammatory response in the wild-type mice, and we're just conserving these changes in physiology in the lung. But interestingly, what we can see is that MECP2 expression is actually not necessarily needed to contain the initial acute response, but really, really crucial to viral clearance by days 10 post-infection. We'll still have the detection of the virus in knockout mice, less so in wild types. And again, at these lower doses, we can recapitulate a decrease in ISG expression at day 6 and 10. At day 6 post-infection, we are now seeing more ISG expression at day 10. And this has to do with, probably, we interpret this to be with virus that still is in the lung, right? But we can see this deep delay in interferon gamma responses. And what we saw in these infected mice at day 10 post-infection is that in the infected lung, there is a decrease in the recruitment of CD8 T cells, particularly activated CD8 T cells, and most importantly, antigen-specific CD8 T cells. When we look at the function of these CD8 T cells, we focused on the two major functions of cytotoxic lymphocytes. The first is the ability to induce cytokine expression. This would be interferon gamma and TNF. This is needed to mediate an antiviral response and kill the infected cells. And we can see that while wild-type mice can have cells that are capable of producing both interferon gamma and TNF, the loss of MECP2 expression actually not only leads to a reduction in cell numbers, but the functionality in their cytokine-producing ability is actually lost. We then look at the cytotoxic potential of these cells to see if there's an uncoupling of these responses. And then what we did here was actually measure both intracellular expression of granzyme B. This is actually the death effector, but also the expression of perforin, or the release of perforin that will make the holes necessary to get granzyme into the infected cell. And we do this by membrane staining of CD107. We saw some of these in earlier responses, and we can see that when we stimulate cells with antigen, we actually lose the cytotoxic potential of these cells. We've done this also with PMA, ionomycin, to bypass antigen sensing to make sure that we didn't have a priming issue, right? And we again see these deep loss. So we think this potentially could be cell intrinsic, but I have to prove that to you. So we went back to the draining lymph nodes, right? Here we're looking at the mediastinal lymph node and try to see if perhaps what was happening is that we're losing the first step of activation and antigen presentation into the naive T cells to drive that differentiation, right? And we did not actually observe that. We could see that loss of MECP2, while it appeared to have maybe a trend of a little bit of a delay in cellular activation, loss of MECP2 did not really affect the emergence of antigen-specific cells in the draining lymph node. So we did not think that perhaps this was a defect in antigen presentation. We think that perhaps then the defect might be in the response to cytokines, which is important not only for the full activation of these cells, but also to guide their proliferation, and also generate that chemokine gradient that's going to push that cell, pull that cell from the lymph node back into the infected lung that I've shown you is already not inflammatory. So now we have a conundrum. If there's no inflammation in the lung, is that the reason it's all cell extrinsic, or do we really have a cell intrinsic deficiency? And so to actually do this, we harness the in vitro stimulation assays that have been developed to study T cell function, in which we can actually take naive splenic cells from wild-type knockout animals, and now we can provide all three activating signals. We can provide antigen and co-stimulatory signal presentation by CD103, by CD3, CD28, antibody stimulation, but we can also provide the signal 3, or that cytokine response, at a controlled manner. We can give both of these cells, both of these cell subsets, the same amount of activating input, and then they should be able to respond equivalently. So the first thing that we looked at was cellular proliferation, and we noted that that was not actually the case. Here we're doing a CFSE dilution assay, where you should expect with each round of replication, you should see dilution of your CFSE, and we see that dye does not quite dilute quite readily in our MECP2 cells, and this is quantified here, where we now see that our cells expand at a much lower rate than the wild-type counterpart, suggestive then a potential cell intrinsic defect. We went back to our favorite assay, the whole genome transcriptome, and actually defined that there's a very distinct response to cellular activation cues in the absence of MECP2, suggesting then that not only might my MECP2 be controlling cell extrinsic factors, it is definitely controlling cell intrinsic signals. And what are these signals? These signals happen to be, when we look at enrichment, happens to be the activation of JAK-STAT signaling pathways, but more importantly, the response to IL-2, which is the predominant signal necessary to proliferate T cells and sustain their viability. We're also seeing some metabolic changes that we also want to follow up on. And lastly, what we saw is that the loss of MECP2 was actually congruent with the aberrant expression of genes that have already been associated to DNA writing and erasing, right? So this is suggestive now that what MECP2 might actually be doing in these cells is being able to interpret those cues. And why we think that this is the case is because it was established more than 10 years ago that the effector differentiation of CD8 T cells actually requires the accrual of new methylation patterns and demethylation of other sites necessary for that effector programming. And the loss of this not only will result in loss of effector function, but also in rewiring of these cells into stem-like cells incapable of responding to antigen. And we can show this here when we look here at demethylated, methylated sites, is that in blue we have the expression of these genes in wild-type cells, their change between naive and effector functions, again in our in vitro assay, and we see that predominantly the demethylation, the expression of genes that have to become demethylated is actually lost. And so with that now, we think that MECP2 leads to a profound deficiency in the lung that makes it susceptible to infection with influenza virus, right? It actually delays the initiation of antiviral and pro-inflammatory responses necessary to mediate these chemokine-mediated migration, or this chemokine gradient. But more importantly, that this susceptibility is also exacerbated by the fact that T cell activation is hindered in a cell-intrinsic manner by the absence of MECP2. And so now, the consequences of this is the loss of alveolar integrity and alveolar function, and then stating that this mouse model is actually potentially telling us that yes, this is a patient population that might have a bona fide, might be bona fide immune compromised, right? And that's we need to be thinking about, how we take this into account for their clinical care. But more importantly, from a mechanistic standpoint, what we're trying to really figure out now is MECP2 actually going back to the issue of dynamics, right? This is not an on-off, right? When are these methylation patterns occurring? Where are these methylation patterns occurring? And do they occur at the same time in all cell types? So will they occur equivalently in the alveolar cells versus the CD8 T cells? How does MECP2 guide the changes in methylation patterns? But more importantly, what I didn't tell you is that MECP2 is one of 7 methyl CpG reading proteins. Does that mean that it is the sole protein that does this? Why don't we see these phenotypes when we actually knock out some of the other methyl CpG reading proteins, and can they actually take over the loss of function of MECP2, and maybe we're releasing an aberrant gene regulation program? So with that, I can go back, switch to Spanish. Como les dije, este es mi grupo. El trabajo que les presenté hoy fue de Matthew McFadden, un estudiante en el laboratorio. Lo hemos hecho en conjunto con profesores de la universidad, con experiencia en respuesta de las células T a los viruses, patología de los viruses, pero también hemos estado comenzando a estudiar un poco más la fisiología pulmonar y la regulación de regeneration of the lung. Pero lo más importante que quiero agradecer es a mi colega Christopher Beatty. Él es el director de la clínica de Rett Syndrome en nuestro hospital local infantil, y a los pacientes con Rett. Han sido muy generosos. Ahora que tenemos esta evidencia en ratones de una deficiencia de linfocitos T, estamos recolectando muestras para tratar de ver, recapitular los estudios que se hicieron hace ya más de 25 años, ver si vemos eso en nuestros pacientes, pero también empezar a ver ahora si hay deficiencia en función y si podemos ver mecanísticamente por qué están ocurriendo estas deficiencias. Y con eso dejo de hablar y atiendo preguntas.
Bueno, tenemos un espacio breve de preguntas. ¿Alguno tiene una pregunta?
A la orden. Yo te escucho y repito. Entonces, a ver, la pregunta fue ¿por qué el pulmón? Y si MECP2 está expresada en todos los tejidos, ¿por qué no otros patógenos y por qué no se presentan esos otros patógenos? Okay. Lo que de pronto yo no les conté es que estos pacientes tienen barreras comprometidas tanto de la piel como del sistema gastrointestinal y del pulmón. Una de las manifestaciones del Rett Syndrome son apneas y dificultades para respirar. Entonces, se sabe que en el modelo de ratón, por ejemplo, se ha usado para definir que se pueden corregir ciertas deficiencias respiratorias si reconstituyes la expresión de MECP2 en las neuronas. Sin embargo, parece que hay en la barrera epitelial funciones metabólicas de MECP2 que no se han estudiado más allá de cuál sería la consecuencia, pero es fatty acid synthesis en homeostasis. ¿Ya? Entonces, Yo creo que aquí lo que está pasando es que no simplemente es una susceptibilidad a patógenos respiratorios, sino que teniendo en cuenta que hay más aspiración, hay más chances de neumonías y se manifiesta mucho más como una respuesta más que todo infección a pulmón. Ahora, para hablar de otros patógenos, usamos un modelo de Staph aureus y esperando que íbamos a ver que simplemente era una respuesta a viruses. Empezamos con viruses porque no se transmiten, o sea, se transmiten por comunidad, no serían parte de la aspiración, ¿cierto? Pero con Staph aureus lo que estamos viendo es, de nuevo, una sensibilidad exquisita a un patógeno que no es letal, un modelo que no es letal en ratón, y con una deficiencia de nuevo en interferón gamma, que es súper importante para contener las enfermedades bacterianas. Y lo que no hemos hecho en estos momentos es definir qué rol pueden tener las células NK, qué rol pueden tener los macrófagos, qué rol pueden tener las CD8 en expresar interferón gamma en ese contexto, porque como dijo alguien antes, entrenado como virólogo primero, pero creo que se manifiesta en todas partes. Adelante.
¿Me escucha? En todo este proceso, la microbiota en el tracto respiratorio, en todo este proceso.
Listo, la pregunta entonces es ¿qué papel juega la microbiota del tracto respiratorio en este proceso? No lo hemos estudiado. Tenemos un nuevo colega que está interesada en la microbiota y sería una pregunta súper interesante que hacer, pero en estos síndromes de autismo lo que se ve mucho es disbiosis gastrointestinal de microbiota, que de hecho está asociada con esos episodios neurológicos. Entonces, hay oportunidad para pensar que la microbiota puede exacerbar muchísimo, o sea, se daña la barrera, exacerbar muchísimo esos problemas neuronales y hacer un triggering del long brain axis que pueda causar estrés, que esté causando señales que suprimen la respuesta antiviral, independiente de, por ejemplo, que las células AT2 estén comprometidas en su capacidad de responder. Ahora, les digo de una vez, ya hicimos el conditional knockdown en las AT2s y aún así, solo con AT2 knockdown, vemos una susceptibilidad súper grande a la influenza. Que de nuevo puede ser la barrera y la microbiota. Y sería súper interesante entonces hacerlo con notobióticos o con antibióticos, ver si se puede mejorar la respuesta.
Agradecemos a la doctora Adriana Forero por su valiosa presentación y por compartirnos con nosotros todos estos aportes.
Accede a este y otros contenidos cuando quieras. Suscríbete, aprende y comparte.